Hello friends, welcome back to my channel BioFrag. Let's talk about DNA polymerases in eukaryotes. So I'll be referring to Pathfinder book. In eukaryotic cells, they contain the numerous type of DNA polymerases in both nucleus as well as the other organelles like mitochondria, and in plants we have chloroplast. And these organelles have their own genomic material that has to be replicated. So for nuclear DNA replication, majorly we have DNA pol alpha, delta, and epsilon. So to start with, DNA pol alpha is an unusual polymerase because it has DNA dependent DNA polymerase activity, and it also has a primase activity, that is 5' to 3' DNA dependent RNA polymerase activity. So this is important for the primer formation because it utilizes the single stranded template DNA and it will form the RNA primers. So it is heterotetramer, and with its primase activity it incorporates nucleoside monophosphates to synthesize the short RNA primers about 8 to 12 nucleotides long, complementary to single-stranded DNA template. These short RNA primers which are formed, they are called as initiator RNA. So on leading strand, it will be just one initiator RNA, and on lagging strand, you will have multiple initiator RNAs, because we know that later they are important for the formation of Okazaki fragment. On extension, you will get the Okazaki fragments, right? So after the initiator RNA is added, with the polymerase activity of pol alpha, this particular pol alpha now adds the 20 to 30 deoxynucleoside monophosphates on the 3' end of initiator RNA. So imagine a lagging strand, single-stranded lagging strand, and it has this multiple initiator RNA, and it will have this open 3' end, to which now the DNA nucleotides are added. Deoxynucleoside monophosphates are added, and it forms the initiator DNA. Now pol alpha does not have an intrinsic proofreading, that is 3' 5' isonuclease activity, and it also has low recessivity. So pol alpha binds to the initiation complex at the origin and synthesizes the short initiator RNA followed by 20 to 30 bases of initiator DNA. Then it is replaced by other DNA polymerases to extend the chain. So first, once the pol alpha's job is done, it is replaced with pol delta or epsilon, and extension occurs. So this replacing is called polymerase switching. Now DNA alpha initiates DNA synthesis on both leading and lagging strands. And replication DNA polymerases or the extension DNA polymerases delta and epsilon, the extended DNA. Now pol delta is multi-subunit nucleo enzyme, and in most specifics it is composed of 4 subunits. It has polymerase activity and proofreading activity as well. So pol delta requires proliferating cell nuclear antigen, which is about 30 kilodalton protein. It is important for its high processivity, and it functions as sliding clamp. So in prokaryotes, we had beta clamps which were acting as sliding clamp. In eukaryotes, we have pCNA, proliferating cell nuclear antigen. Functional pCNA is homotrimer and acts as processivity factor. It also requires a loader, clamp loader, that is replication factor C here, and there it was the gamma complex in prokaryotes. In eukaryotes, we have replication factor C acting as clamp loader. Now talking about DNA pol epsilon, it is a multi-subunit nuclear enzyme composed of 4 subunits again, and it has both polymerase activity and proofreading activity. It is the most accurate eukaryotic DNA polymerase and able to carry out high precessive DNA synthesis. Highly precessive DNA synthesis with the aid of sliding clamp and loader. So it also requires clamp loaders and clamp sliding clamp. So it was previously thought that epsilon's main function was to extend the leading strand. during the replication. But now, according to the recent study, what is suggested is that Paul delta alone replicates leading and lagging strength. Replicates as in extends leading and lagging strength. So we cannot really define, the precise role of Paul epsilon here, but it is somehow involved in replication. Paul delta. DNA Paul gamma is sole polymerase participating in mitochondrial DNA polymerase. Very important. In Homo sapiens, it is composed of three subunits. Both it has polymerase activity and proofreading activity, and there is this inhibitor, that is FED-corin. Okay, this FED-corin inhibits all alpha, epsilon, and delta, okay, mammalian nuclear DNA polymerases, but somehow it does not affect the mitochondrial DNA polymerase, that is Paul gamma. So to summarize, we know that all these polymerases, they are polymerases, right? DNA polymerases. So they all have 5' to 3' polymerase activity. But Paul alpha does not have both the exonuclease activity. None of this, okay, have 5' to 3' exonuclease activity. Paul alpha and beta do not have 3' to 5' proofreading activity. But gamma, delta, and epsilon have proofreading activity. Gamma is the sole polymerase involved in mitochondrial DNA replication. And this particular delta is involved in leading and lagging strand synthesis, and beta, although we didn't speak about it much in this video, it is involved in base excision repair, so repairing the damaged DNA. And Paul alpha, as we all know, it has primase activity, so priming during the replication is done by Paul alpha. So that's all about the eukaryotic polymerases in this video. If it was helpful, please like, share, and subscribe the channel. We'll be looking at the replication process in detail in upcoming videos.